my dear little chutku people, I hope that you are doing as wonderful as you all are, and I welcome you all to PU2 Brahmastra Series, where today we are going to talk about biology, and the chapter's name is Human Health and Diseases. Now, why did ma'am choose this chapter? Because it carries 11 marks for you. Why is it not coming? Yes. It carries 11 marks for your PU2 board exam just after your molecular basis of inheritance and principles of inheritance and variation, which carries 13 marks. Over here we have a 11 marker chapter. So can we ditch this chapter by any chance? No, we cannot ditch this chapter. We have to study this, yeah. And we have 3 blueprints. According to blueprint 1, there will be 3 questions, that is 1 marker questions. There will be 1 3 marker question and 1 5 marker question. According to blueprint 2, there will be 1 question from each category, 1 marker, 2 marker, 3 marker and 5 marker. Making it a total of 11 marks. Okay. According to blueprint 3, which is the most disastrous one, there will be 1 question from 1 marker. There will be 2 questions from 5 markers. So your 10 marks you'll get here itself. So we have to be prepared for all type of questions this time. 2 marker, 1 marker, 3 marker and 5 marker. So according to it, I have made sets of questions. There will be 15 MCQs that we will be solving today, because anyways, you see, MCQs will be there for sure. Okay, there will be 2 marker, because 2 marker is just at one place, you see that's why 2 marker, and there will be 5 3 marker, because you saw it at 2 place, that there are chances that 3 marker could come, and then there are 6 5 marker questions. Works? So let's get started. Before that, for the people who are asking whether, ma'am, if we study this, do you think everything will be, we can complete the whole chapter? Yes, I think that you can definitely at least cover the most important topics, and I'll be definitely telling you the important topics at the end, which you can also refer in an extra way. Okay, from the NCERT, nowhere else. Okay. Works? So are we in this journey? If you are in this journey, say yes ma'am, we are in this journey. I really get happy when I see you talking to me during the live classes. Okay, chalo. And one more thing, if you want to know the schedule for the week, this is the schedule. Today we are here, Jasmine Ma'am is going to take human health and diseases. After this, there will be Aishwarya Ma'am doing chemistry, and then physics, and then math with bio, and then math again. So you have the timings, you have everything. If you want to take the screenshot, you can take it. Okay, and don't take this particular series lightly. This is very important series, I'm telling you. So first question from your model question paper which has come: interferons are most effective in making non-infected cells resistant against the spread of which of the following diseases in humans? So the question says, okay, if some cell is releasing something which is known as interferons, these interferons would be helpful for some cell, because these interferons are the one which makes this particular cell resistant against some type of pathogen, and they are asking you which would be that pathogen and which is that particular disease. So interferons basically would be released only if the cell is infected by some virus. For example, this particular cell is infected by HIV virus, okay, and now this cell will be releasing what? Interferons. These interferons would be alarming other cell that, hey, you have to be resistant, there is a virus attack over here, you have to be safe. Okay, so basically the question is, interferons are most effective in making non-infected cell resistant against the spread of which of the following diseases? It has to be a viral disease. So among this, there is only one viral disease, which is what? AIDS. I hope you got it. Okay, just remember, interferons are released only if the cell is infected by virus, and now it is going to alarm other cell to be staying safe from the virus. Cool? So this is the answer for that. Going to the next question. The human host cells in which the gametocytes of malarial parasite develop are. So basically, listen, malarial parasite, what is a malarial parasite? It is known as Plasmodium. So Plasmodium, basically, you can remember like this. They have their kits inside. What say? Inside the mosquito. Inside the mosquito, which is Anopheles mosquito. Actually, they do not have kits. Basically, there is larval forms that are being made. But anyways, the female and the male gametes comes to fuse, and everything happens only inside the mosquito. Okay, and now the mosquito will be making something that is known as sporozoite. Okay, something that is known as sporozoite, which it gives to us when it bites us. So in our body we have sporozoites. Initially where, children? Where does this sporozoite go? It goes to the liver. It goes to the liver, divide itself, cause whole ruckus there, damages a lot of cell, and then slowly it will be moving to the next area, that is RBC or red blood cell. In the red blood cell, they will be transforming themselves into next type of cells which are known as gametocytes. So gametocyte is a stage. Okay, gametocyte is a stage. In this particular stage, then the next thing that they will do is form gametes. But over here, in the case of human body, they are not going to make the gametes. You know, I told you the kits are being formed over here. So basically, gametocyte stage is reached over here in the RBC. Now, when one mosquito drinks our blood, it is taking in the RBC, which has what? The gametocytes. Now, the gametocytes later enter the mosquito's body. In the mosquito's body, again, they will undergo sexual reproduction and then form sporozoites. Again, this guy comes and bites us, and the same cycle repeats again and again and again and again and again. So, the human host cells in which gametocytes of malarial parasite develop are, which is that cell where the gametocytes are being developing? It is what? The erythrocyte, that is RBC. Okay, liver cells, you call them hepatocytes. In liver cells, they don't form gametocytes. Okay, the next stage of development comes only in the RBC. But in RBC also, they won't be forming gametes. The gametocytes are being formed. The gametes will be formed only once it enters the mosquito's body. Inside the mosquito's gut, they will be forming gametes. The gametes fuses, later forms some certain things. There are so many stages. At the end, you have sporozoite being formed. And this sporozoite is given back to us. It goes to liver and infects this. Works? Okay. Chalo, next question. A farmer working in a field was bitten by poisonous snake. Okay. Doctor gave him an anti-venom treatment. What did doctor give children? Anti-venom treatment. That contained preformed antibodies. As in, it already had certain antibodies. So usually what happens, I'll tell you. For example, in our body something happens, okay, or some pathogen gets in. When some pathogen gets in, this pathogen will be having certain antigens on the top of it, certain proteins which are very foreign to us. What do you call this? Antigens. And then our body, when it sees this particular antigen, they will be releasing certain antibodies. Okay, certain antibodies on the basis of this, and these antibodies will later call other cells and try to kill this particular cell in a whole. But over here, we have to wait for the antibodies to recognize these antigens and do things like that. Now, for a person who is bitten by a snake, can they wait for their body to produce the antibodies? No, children. It's a very slow process. We cannot depend on that. What about giving them the antibodies directly? What about not waiting for the antibodies to be released by the body? Instead, what if you give them the preformed antibody? It will be a really great thing. Now, this type of immunity where you are ready-madely providing antibodies over there to them so that they can be safe in several minutes or seconds, this is known as passive immunization. What do you call it? Passive immunization. Okay, in our body, when it happens naturally, the antibodies are released. You call it active immunity. Our body is actively participating in producing antibodies, but this is a very slow process. We cannot rely on that as of now. Okay, so in anti-venom treatment, they give preformed antibodies, and this is an example of passive immunization. I hope it helps. Okay, and there is one more thing. There is vaccination as well. There is vaccination as well. What happens in vaccination? In vaccination, we basically provide the body with dead microbes, or probably live attenuated. Live attenuated as in, we are disarming the pathogen. The pathogen cannot cause any trouble in us. They just have the certain proteins which is foreign to us. That's all. Okay, such that we are alarming our body. In a way, we are alarming our body and saying that, okay, this antigen is there. Please prepare certain antibodies and keep it. Please prepare certain antibodies and keep it. The next time the actual version of this particular bacteria or whatever pathogen comes, you have to release these antibodies. This is what you mean by vaccination. Okay, that is what you mean by vaccination, where you are allowing your body itself to release so many antibodies. Okay, but you're giving enough time also. For example, in Covaxin, Covishield, what were we doing, children? We were providing the, the dead form, the attenuated form of the virus. Okay, some spike proteins were there. Now our body knows that, okay, this is something foreign and they have to produce antibodies. So they definitely produce antibodies, which is why we got the fever and things like that. Later on, when actually this infection comes, our body will fight back because it already has certain antibodies being made. So this is next thing that we have apart from passive immunization. So passive immunization is something that we do when there is a quick need of things. Okay, when there is a quick need of things, we do passive immunization. When the things can be taken slowly, we do vaccination, where we rely on our body only. Okay, works? Next thing. Nowadays, diseases like dengue and chikungunya are widespread in different parts of India. To prevent the spread of the disease, which vector has to be eliminated? Vector is what, children? Vector is an organism which would be transporting the pathogen. Okay, vector is something that will be transporting things from A to B. So, for example, here the vector will be thing that will be transporting things from some other places to human beings or any whatever organism it is going to affect. So, over here, what is the vector that is being asked? In dengue and chikungunya, who is that organism which spreads the disease? Is it Culex mosquito? Is it Anopheles mosquito? Is it female Anopheles, or it is Aedes? I'll say both dengue and chikungunya are being formed by Aedes mosquito. It is formed by Aedes mosquito. Culex mosquito is there for transporting, for bringing out one disease which is known as, can you tell me in the comment section, what does Culex mosquito do? The disease's name starts from F. The disease's name starts from F. Can you try it out? It's very easy. Now, Anopheles mosquito, they have given the option. Anopheles mosquito is there for transporting or causing this particular disease which is known as malaria. Okay, and over here female Anopheles mosquito is given. Obviously, it's always the female mosquitoes that transfers the disease. Please remember this fact that it is the female mosquitoes that transfers the disease, because female mosquito goes and feed the blood. The male mosquito feeds on the plant sap. It does not feed our blood. Please remember that. Okay, so Aedes mosquito also over here, it's a female one. Cool. So, it's a Aedes mosquito transporting this and this. Next, the disease chikungunya is transmitted by, again the question came, house flies. This question have come in model question paper this year, okay, and next 2023 March also this question has come. The disease chikungunya is transmitted by, tell me, house flies, Aedes mosquito, cockroach, female Anopheles. It will be what? Aedes mosquito. See? Now next thing, elephantiasis is caused by, okay, I told you the disease's name that starts from F, it also starts from E. There are two names. Okay, anyways, elephantiasis is caused by something which is known as roundworm, filarial worm, ringworm, plasmodium. What is the answer? Elephantiasis is also known as filariasis. What do you call it? You also call it filariasis, my dear children, and it is caused by filarial worm. But I want to bring out your attention over here. Okay, filarial worm. Okay, basically, how do we get the filarial worm? There will be a vector that will be transporting the larvae of this particular worm. And who is that vector, children? That vector is Culex mosquito. So, those of you got the answer correctly, good work. Okay, I will be appreciating you in the comment section only. I will be telling you in the live because I will be also attending the live. Okay, so I will be putting down there. So anyways, filarial worm that you see over here, it is spread with the help of Culex mosquito. Culex mosquito carries the larvae, it comes and bites us, and this larvae gets into our body, grows into our body, forms the filarial worm, forms blood clots, lot of trouble happens in the lymphatic system such that the person's lower limb, it becomes very, very big, it becomes swollen. Okay, the genital organs, the lower limbs, everything becomes swollen, which is why it is known as elephantiasis. There is elephant-like legs. Okay, you got it? Works? Chalo, next thing. Identify the wrongly matched pair. Tell me the wrongly matched pair, and I'll just turn on the AC a bit more because it's too hot up here. Yes. So, common cold is caused by Haemophilus influenzae. Is it so? Is it so? Guys, with the help of this, you have to remember the causative organism and the disease. Please, that's why I put this question up here. So, common cold is caused by Haemophilus influenzae? No. Haemophilus influenzae causes something that is known as pneumonia. What do you call it? Pneumonia. And common cold is caused by something that is known as rhinoviruses. Okay. Now, pneumonia is caused by Streptococcus pneumoniae. Is it correct or it is false? It is correct. Amoebiasis is caused by Antamoeba histolytica. It is correct. Ringworm is caused by Trichophyton species. Yes, it is caused by Trichophyton, Epidermophyton, or what do you call it? Microsporum. There are so many things. There are so many fungi being involved over here. So, this is the fungal disease. This is the protozoan disease, and this is caused by a bacteria, that is Streptococcus pneumoniae, and also Haemophilus influenzae. Now, common cold is caused by rhinoviruses. This is the wrong option. So, basically, a wrongly matched pair. Okay, wrongly matched pair. This is not the correct pair, which is why this becomes your answer. Works? Chalo, next thing. Which group of symptoms are correctly matched with the diseases? So now you have to be looking for the symptoms. Okay, why did I put this question up here? Because there are so many questions that you will be seeing in 5 marker also. They have asked about the name of the causative agents and stuff like that. So you cannot miss this information. This is very important for all the diseases. You should be knowing the causative organism and the symptom. Cool. Okay, so malaria, high sustained fever, is it there? Okay, weakness, loss of appetite, constipation. Constipation is not there in malaria. Okay, fever also, it is not sustained fever, you get it in bouts. Okay, initially just for a second you will get too much fever, you will shiver, and then it goes. So basically this is wrong. Next, pneumonia, nasal congestion and discharge, cough, sore throat and headache. Basically cough is there, you will be having headache also. Talking about nasal congestion and discharge, it is not very common in pneumonia. In pneumonia, you won't have discharge. Usually, it is there in common cold. Okay, usually it is there in common cold. This particular thing, nasal congestion and discharge is found in common cold more often. Okay, and sore throat also is found in common cold more often. In pneumonia, what are the symptoms that you can find? You will be having cough, you will be having headache, you could get fever, and also the thing is, if the pneumonia it is increasing way too much, your lips will turn different in color and your nails will also turn different in color. It will be blue because of oxygen deficiency that is being caused over there. Okay, now amoebiasis, in amoebiasis where basically your intestine is getting affected, over there it is caused by ant amoeba histolytica. What is it caused by? It is caused by ant amoeba histolytica. Ma'am, how do you remember these things? Basically, if we do it again and again, if we go through it again and again, you will be able to remember. I am telling you. Okay, trust me, I am also a student just like you. Okay, you can think like that. So amoebiasis causes constipation, abdominal cramps, blood clots. Yes, amoebiasis, always remember, usually people may have dysentery, loose motion. In loose motion, what's the difference between loose motion and dysentery? In loose motion there is no blood. In dysentery there will be blood also along with the stool. So basically amoebiasis is also known as amoebic dysentery. Over here you always have to remember there will be blood loss and things like that happening through the dysentery. So constipation, abdominal cramps, blood clots, it's happening, and this is the correct answer. This is the correct match. Let's see common cold also. Common cold, difficulty in respiration, fever, chills, cough and headache. Difficulty in respiration you would not find in that much level in the case of common cold. It is not a disease that affects your lungs. Please remember that. Common cold is not something that affects your lungs. It affects your airways over here. Okay, pneumonia is that something that affects your lungs. So your lower respiratory tract is affected by pneumonia. Your higher respiratory tract is affected by common cold. Okay, pneumonia causes trouble in both, but this guy is only for the upper respiratory tract. So difficulty in respiration does not comes under the chills and all. No, children, there is no chills and things like that you can find over here. Okay, got it? So amoebiasis has the correct option. Now if you are not aware about the symptoms, will you be able to do this answer? Which is why I say you have to try. Now, which of the following diseases are vector borne? Vector borne as in, for example, some organism is coming in between. Some organism is coming in between and causing this particular disease, or basically it is carrying some stage of that particular pathogen. Okay, please remember there are two things mentioned in your NCERT. So one thing that they have mentioned is vector, and the other thing that they have mentioned is mechanical carrier. Mechanical carrier as in it is just carrying the content. Whatever larvae, whatever is there, it is just carrying stuff. For example, in amoebic dysentery, house flies are there that carries the contaminated stuff and then goes sit on the food and then it gets. It is not getting, in its body there is no stage of the pathogen going on. Okay, but when you talk about a vector, you will be finding that some stage of the pathogen is being spent inside the vector as well. And now this vector will be coming and biting us or doing something, basically transferring that content to us as well. For example, in the case of malaria, you saw plasmodium. Plasmodium has the gametocytes within it. They fuse, they show the reproductive stage is happening in there. Okay, so you saw that. Culeus mosquito also has the same thing. So they are all acting as vectors there. Okay, the mosquitoes are acting as vectors there. So amoebic dysentery, ringworm, this is not something that is a vector-borne disease. Okay. Now filariasis or filarians and pneumonia, again it's not vector-borne. This is airborne, or basically, yeah, it is airborne thing. Okay. Typhoid, ascariasis, this is Basically, typhoid is caused because of contaminated food and things like that. Same with ascariasis as well. Over here, food and water, whatever is caused, it causes things. Malaria and elephantiasis, this is something that is vector borne. Malaria by plasmodium. Malaria by plasmodium, but what is the vector, children? The vector is your Anopheles mosquito. Okay, elephantiasis caused by filarial worm. And what is the vector, guys? It is the Culex mosquito. Got it? So these are the vector borne diseases. So this becomes your answer. Works. Chalo, next question. Identify the type of antibody and the type of immunity provided by colostrum to newborn. So colostrum is what? The initial milk that the mother gives during lactation. So it will be yellow in color. So in this yellow milk, what type of immunity is provided to the newborn with the help of this yellow milk? Basically, I'll say over here, colostrum is rich in one type of antibody, and that antibody is A. IgA, antibody is also known as immunoglobulin. It's a protein, right? So immunoglobulin A is the antibody which is very much rich in the blood, in the milk, breast milk. Okay, always remember A is the one that is there in the milk. How can you remember? So remember this thing, gamed. These are the antibodies. There is IgG, IgA, IgM, IgE, IgD. Okay, G comes first in gamed, right? The G is the one that is being transferred first from the mother to the newborn with the help of placenta. IgA is the one which is transferred with the help of milk. Then the others, basically the baby will be acquiring it later. With time, it will be forming in its body slowly. Okay, so placenta and milk, you have to remember it. So basically, IgA is being given from the mother to the newborn through the colostrum. Now over here, is the baby making its own antibody? No, the mother is providing. In antivenom case, that point also, you saw the antibodies are ready-madely given. Over here, this type of immunity where our body is not actively participating in the production of antibodies, what do you call it? You call it passive immunity. So IgA and passive immunity becomes the correct answer. Works? This is question number 10. Okay? Next question. A person who undergoes organ transplantation would have the fear of graft rejection. Identify the type of leukocytes involved here. So I want to tell you one very important thing. Okay, on every cell of our body, there are certain proteins which are known as MHC proteins, major histocompatibility complex. And this is unique to us. This is very much unique to us, just like our fingerprints. So for example, I am getting a graft from my father or any other person. Still, when my body cells sees that this particular MHC or major histocompatibility complex is different from mine, what will happen? Basically, my body will be fighting against this, thinking that this is something foreign. I should not be letting it live inside my body, and I'll try to kill it. This is known as graft rejection. This is known as graft rejection. So usually it happens when we are totally unrelated. Okay, more in it can happen anytime, but it is more common when people who have no relation between them, they try to exchange things. Okay, so there the MHC basically is thought to be something different, and since it is very foreign, the body tries to kill it. But which are the cells which actually finds and kill them? Basically, it's a type of T lymphocytes. They are what? T lymphocytes which comes to action. T lymphocytes has the capacity to check the MHC molecules, the MHC things that are present over here. T lymphocytes, in T lymphocyte there is T cytotoxic lymphocytes, which is the one which has the ability to check the MHCs which are present, and depending on it, it will go and directly kill. Okay, so a person who undergoes organ transplantation would have the fear of graft rejection. Identify the type of leukocyte. Leukocyte as in WBCs, which are involved over here. So you have B lymphocytes, which is WBC, T lymphocyte, again a WBC, monocyte, again a WBC, neutrophil, again a WBC. But which one comes in graft rejection? It is T lymphocytes. Specifically, which one? T cytotoxic cells, TC cells. So there are two cells, T cells, one is TC, other one is TH. So TC cell are the major ones that comes into play. Works? Next. Identify the enzyme which deviates from the central dogma of genetics during HIV infection. What do you mean by central dogma of genetics? Central dogma says from the DNA, you will be making the mRNA. This is known as transcription. Now from the mRNA, you will be making a protein. With the help of the mRNA, you will be making a protein. What do you call this? This as translation. This is known as the central dogma. That is basically DNA to DNA if you want to form, what do you follow? You follow replication. So there is replication to form enough amount of DNA. If you want to form mRNA, you do transcription. If you want to make protein out of it, you do translation. This goes in step-by-step order. This is known as central dogma. Okay. Now, identify the enzyme which deviates from central dogma. There is some enzyme which will be helping the virus to deviate from this. So what does the virus do? No, virus has usually RNA as the genetic material. Virus has RNA as a genetic material. Now it usually goes and infects organism that has DNA as the genetic material. So virus itself, it will be having RNA as a genetic material and it is going to infect organism that has DNA as a genetic material. So to not being caught, what do is the RNA which they have, they are going to reverse transcribe. This is known as transcription. If I am reversing this process, what do I call it? Reverse transcription, where with the help of RNA I am going to make a DNA, and this DNA I am going to put inside the human DNA and make its copy. So my DNA, my human DNA won't be identifying this, and my DNA will be making copies for it also. This is known as reverse transcription, and this is done with the help of an enzyme which is known as reverse transcriptase. Okay, what do you call? Reverse transcriptase. Got it? Chalo. Next question. Cancerous cells continue to divide, giving rise to mass of cells called tumor. The property responsible for this is what? So cancer cells, it continues to divide. That is something that is catchy over here. It continues to divide. So what happens to normal cell and what happens in the case of cancer cells? So cancer cells, okay, normal cells I'll make first. For example, this is a box. I have one cell. This cell divides into two. Other cells divides. Again, here it will be stopping. Here basically it will be stopping. When it touches the other cell, it will be stopping to divide. It won't go on further dividing. But when you talk about, and this feature is known as contact inhibition. When it comes in contact, it will inhibit itself from dividing. Okay, this is known as contact inhibition. In the case of cancer cells, for example, this is the box. It divides, it keeps on dividing even after being in contact with each other. Divides, divide. It will keep on dividing and come out of the box as well. This is known as loss of contact inhibition. They lose this property of contact inhibition. So that is what is given over here, and this becomes your answer. Works? I hope children you are understanding. If you are understanding, do let me know that okay ma'am, we are understanding, it's fine. Okay? And if you are not understanding, then also you can tell me. Okay? If you have doubts, put it down here, I'll solve it. Cool? Now, immune system elicits a highly intensified response when it encounters a pathogen subsequently. It is due to what? So, immune system, it elicits as in it causes a highly intensified response. Highly intensified as in it brings out so many cells in this particular response. When it encounters a pathogen subsequently, as in the next time, the second time it encounters a pathogen, it gives out a big response. It is due to what? It is due to what? Primary response, secondary response, tertiary response, or both. So basically what happens is that first time an organism sees a pathogen, at this point of time it might not be able to fight back the disease, which is why we get diseased. Okay, we all might have got coronavirus once in our life. The second time the same pathogen comes, okay, our body will be fighting back against this. Our body will be fighting back against this. So the first response that comes is known as primary response, my dear children, and the second time it is responding, it will be known as secondary response. And in secondary response, this will be a more intensified response. This time our body is prepared, it knows what antibodies have to be made. This time our body will fight back really well. Okay, so in primary response, there are two types of cells being made. Okay, one is antibodies and other is memory cells. One is antibody and the other one is what? Memory cells. This memory cells have the memory of things happening. Next time something like this comes, it will be immediately forming that antibodies which it made now. Okay, so that is why I say secondary response is the one that has more intensity than the primary one. Cool. Next, last MCQ. Identify the disease and the test suggested for the same based on the characteristic symptom. Okay, so we have to identify the disease and also we have to see the test. So intestinal perforation, typhoid and Vidal test. In typhoid, do you think intestinal perforation will be there? Yes, typhoid is a disease that will be affecting your intestine, and if it is going unseen, okay, it will lead to intestinal perforation that would lead to death also. Intestinal perforation as in there will be holes in the intestine, okay, and this is tested, okay, with the help of Vidal test. So this is also true. So first point itself is true, but let's see the other. Severe chills, profuse sweating, ascariasis, no children. This will be malaria, and malaria does not have a test like not given in your textbook. It's not ELISA. ELISA is for viral infected diseases. That's AIDS. Bouts of fever, unexplained weight loss, no children. Blockage of intestinal passage, nausea, typhoid, no children. It is not typhoid. It can be it can be ascariasis. Okay, and it is not said by ELISA. So this becomes your answer. Cool. So that's why I said symptoms are very, very important. Please learn it very, very, very much. Okay, so now let's just have a quick break for water and come back right now. Okay, I will also have my water. This is a reminder for call for you to have your glass of water, okay, or your bottle of water. Just give me a second. I'm coming in a second. Okay, so water break is done. Let's see. Draw the structure of an antibody molecule. This is a model model paper question. Okay, so 2 marks. So how can you draw this structure of an antibody? Please remember, you have 2 strands, okay? One strand goes like this, the other strand goes like this. This is known as, these 2 things that you see over here, they are known as heavy chain. They are known as heavy chain. And then comes 2 smaller strand, okay? 2 smaller strand that you see over here, which is known as light chain. They are small, no? So I will say light chain. Cool. Now heavy chain and the light chain has a specific region over here, okay, which is different for different antibodies. So over here, this region that you see, okay, this region that you see, it is known as variable region. It is different. It is known as variable region, whereas this and this thing that you see over here, it is known as constant region. What is it known as? It is known as constant region. Now the heavy chains, they have to be bound to each other, so they have disulfide bonds. What do they have, children? Disulfide bonds over here, so that both of them, they are together. Similarly, in between the light and heavy chain, they also have sulfide bonds. Cool. They have sulfide bonds among themselves also like this. Please remember that as well. Okay. Now this antibody will be having a site for antigen to come and attach, or basically for it to go and attach to an antigen. Okay. There will be a site over here which will be allowing it to attach. The site is kind of specific for all. What do you call this particular site as? You call this as paratope. Okay, remember that these are the legs. Pair, in Hindi we say no, pair. So this is the paratope. On the antigen also, they will be having certain specific areas like this. Okay, this will be known as epitope, which will be coming and binding over here. Fine. Now this is a normal structure, but I'll recommend you to draw something like this, where there's just double things. Okay, instead of one strand, they made a bit thicker one over here. I didn't make that thicker one, but you can make it. So the same thing that I was trying to say over here, this is the heavy chain. Okay, this whole thing that you see is the heavy chain. Then you have the light chain. Okay, in the heavy chain and light chain, you have this region which is what? Variable region, and this region that you see over here, it is constant. If you want, I can mark it over here for your understanding. So this thing that you see, this is constant region, both here and here. This thing that you see, it is the variable region. Now over here you see they have sulfide bonds among themselves and also internal sulfide bonds. Okay, here also between the light and heavy chain, they have sulfide bond, just one some sulfide bond over here. Okay, internally they have so many, that is different. Cool. And there will be an antigen binding site, and in antigen also there will be an antibody binding site, which is known as epitope. Works. So this is the structure of antibody molecule, which is very, very, very important question. Came in your model question paper, came in your other previous year questions as well. So have this structure really in your mind. Okay, works? Chalo, next thing. List any 2 methods of HIV transmission. So HIV is a virus, okay, is a virus, human immunodeficiency virus, and it is transmitted with the help of body fluids. With the help of body fluids. So what all time do you think the body fluids can be exchanged? That's the time when we can say that HIV has spread. For example, 2 people having sexual intercourse. One person has HIV, the other person will also get HIV virus. Okay, because it is transmitted through the body fluids, and you know the seminal fluids, genital fluids which is being released is also a type of body fluid. So sexual intercourse is one such thing. Now, if someone is a drug abuser, someone who uses drug a lot, and which type of drug? Intravenous. Okay, so for example, me and my friend is taking drugs with the help of injection. That friend has HIV. Now I am taking the drug with the help of same needle I am using. So isn't it the blood is being in contact, right? In this way also, basically what happens, children, HIV is spread. There are 2 things that they asked. I have given you 2 things, but apart from this, I will tell you next thing. Okay, blood transfusion, unchecked blood transfusion. For example, a patient gave blood, but that patient did know that that person has HIV, because HIV is not something, AIDS is not something that will show you the symptoms very soon. It is a very slow process that the symptoms develop. Okay, so basically this person gave the blood, and the people who collected it didn't check what type of blood it was, whether it was HIV positive or not, and then they gave it to the next person or patient who was in need of the blood. Probably people who had thalassemia and things like that, who need repeated transfusion of blood. At this point of time, unknowingly you are basically transporting this particular blood, and hence you are giving this, or you are basically spreading this particular disease. So blood transfusions is one big reason. Okay, now here are the answers. You can see sexual contact with infected person, transfusion of contaminated blood and the blood products, sharing needles as intravenous drug users, from infected mother to the fetus through the placenta. To the placenta also. It is very rare things to go like that. Blood on blood usually does not matter, but somehow if there are certain bruises, anything in the child or anything like that, the blood is coming in contact, the my dear children, it will be infecting. So infected mother to the fetus also, it's possible with the help of placenta. Now organ transplantation from an infected donor to the recipient, that is also one reason. So in your books, these are the things that is being mentioned, so you have to remember it. This came in July 2017. So these are the few methods of HIV transmission. For 2 marker, you can write the 2 points, or probably you can go write 4 points as well. Cool? Works? So next thing, name the drugs, its source and nature given in this table below. Find A, B and C. So over here, the name of the drug you have to say. Source plant is Papaver somniferum. It is a depressant. So Papaver somniferum is the first type of drug that is being mentioned in your book. Okay, they gave rise to opioids. From here, you get opioids. Papaver somniferum is the plant that will be giving you opioids. In opioid come two of these things which are given in NCERT. One is heroin and the other one is morphine. So morphine is used in the case of hospitals, okay, because morphine acts as painkiller and sedative. Morphine is used for acting as painkiller and sedative for people to sleep properly. So people who have very big operations and things like that, where it's too much difficult for them to handle pain at this point of time, morphine is given, but it is under regulation. Okay, then heroin is a drug. It is a different form of diacetyl form of morphine. Okay, but it is being produced from the same plant, that is Papaver somniferum. Little bit amount of more changes are being done for heroin, and heroin is the one that acts as a depressant. It acts as a depressant, my dear children. Okay, so please remember opioids are the one, examples heroin and morphine, and over here morphine acts as a painkiller and sedative and heroin acts as a depressant. Opioids are the one that is basically affect your central nervous system. I am trying to explain you the drugs here itself so that you don't find it difficult later on. And it also brings about difficulty in the gastrointestinal tract, as in your gastric tract. Okay, so central nervous system and gastric tract, both of them it affects. And the name of the drug over here which is acting as a depressant will be what, children? It will be heroin. Okay, cool. And you get it from the latex of opium poppy, or basically Papaver somniferum, latex, from the milky thing. Okay, you get this particular drug. Now cannabinoids source plant is this, and effects on cardiovascular system. So over here it was affecting on the central nervous system and gastrointestinal tract. Remember, this is the only drug given in your book which is acting on the cardiovascular system. So cannabinoids come under this category. If you have heard about cannabis, this is an example, and the name of the plant is cannabis sativa. Where shall I write? Cannabis sativa. I have written it properly elsewhere, you can go read that as well. So examples of this is ganja, marijuana and things like that. Okay, all the different parts of this particular plant can be used for forming different drugs, and all of them, they have effect on the cardiovascular system, and also sometimes it brings out hallucination, which is not given in your NCERT, but I am telling you. Okay, now cocaine is the next type. Cocaine is, you get it from erythroxylum coca, and what does it do? Is it a depressant effect on cardiovascular system? What does it do, children? It is, I told you, there is only one guy which affects the cardiovascular system. All the other guys, they are going to affect your central nervous system. So it is going to have effects on CNS, or basically central nervous system. And cocaine basically gives a sense of euphoria or unlimited happiness, because it goes and brings about pathway block of dopamine. Dopamine is a happy hormone. So dopamine, usually when it is being released from one neuron to the next, it goes and then excites the other one. Okay, but what happens that later, after a point of time, it goes back to the previous neuron. But over here you block that pathway, so it is kept on releasing. Dopamine is kept on releasing in a way that it does not stop, which gives a sense of happiness, euphoria, okay, and hallucination as well. So do not try it at home, but yeah. Okay, so this is something that you need to remember, and please try to remember it, and this is how you can write the answer. So A is heroin. Cannabis sativa is the name of the plant that gives you cannabinoids. All the different parts of the plant can be used for ganja, marijuana and things like that. Effects on the cardiovascular system. Cocaine, it comes from coca alkaloids. Basically you get it from erythroxylum coca. You have to remember the name of the plants. Okay, and then the nature is, it affects your central nervous system just like them. Just like them, it is also going to affect your central nervous system. There it is acting as depressant. Over here, you remember, it is a happy one. Okay, but that does not mean you should be having it. Okay, so this is a model question, so you have to be really careful over here. Go through it once more. Okay, next, describe any 3 properties of cancerous cell. Can you tell 3 properties of cancerous cell? One property we already told. Loss of what? Contact inhibition. So when it comes in contact, it stops dividing, but over here it will keep on dividing. So loss of contact inhibition. Uncontrolled division is also one thing. Usually what happens, the cells, when it divides, it has a span. After few sets of division, it will stop. It will stop dividing, my children. So we say that is controlled division. Over here you will be finding something that is known as uncontrolled division, my dear children. Okay, there will be uncontrolled division. Since there is a loss of contact inhibition, so I can make into 2 points, 1 and 2. Loss of contact inhibition, and the next point is cancerous cells show something that is known as metastasis. Metastasis. What do you mean by metastasis, my dear children? For example, here I have certain cancerous cells. These guys will be moving to other areas of my body. Okay, it will keep on spreading to different locations of my body, which is why we say the cancer becomes more grave here when it comes to different stages. So after some stage, it will show metastasis. It will keep on going to different parts of the body, which is kind of very dangerous. So these are very important 3 properties of cancerous cells, which is written over here. Uncontrolled growth, they divide rapidly without responding to normal regulatory signals. So there are signals in our body which controls things, but over here they don't listen to anything. Okay, metastasis, as in they can spread to distance of the body part, distant body parts via the blood or the lymph. Loss of contact inhibition, they grow even when in contact with other cells, forming what? Tumors. Okay, cool. So this is a 3-marker question. We have started off with 3-marker question. So these are the 3 properties. So it's a very direct question that has come. So I hope you're getting the answer. I'll tell you one very important thing. Cancer is a very important topic. There are so many questions around cancer. First, one question could be properties of cancer. Second question will be treatment method available for cancer. Third question will be detection of cancer. Fourth will be what are the causative agents of cancer. So the topic itself is so very important. So please go through it. Okay. Next. Name the pathogen, vector and the symptom of malaria. Okay, pathogen, tell me. You see how many times malaria has been repeated? So pathogen's name is Plasmodium. There are different type of Plasmodium. Plasmodium vivax is there. Okay, apart from that, can you tell me a name? Can you tell me the name of the most malignant type of malarial form, vector or pathogen? Can you tell me the malignant as in the pathogen which causes the most amount of ruckus? Who is that? Plasmodium vivax, then comes what? Plasmodium malariae. Okay, that does not cause much trouble. M should be small letter because this is a species name, okay? And then the malignant form is falciparum. Malignant one is over here. Okay, it causes a huge lot of trouble. Now, what is a vector of malaria, children? It is the female Anopheles mosquito. Okay, and what is a symptom of malaria? You will be having fever, you will be having chills, you will be having sweating. All this are features of, or what, symptoms of malaria. So over here I have written everything over here. So see, Plasmodium, P. vivax, P. malariae and P. falciparum is the pathogen. Okay. Now vector, by biting of infected female Anopheles mosquito. Symptoms, high fever, chill, fever occurs on every alternate day, vomiting is there. Okay. So please remember, it's a very important thing how you have to frame your answer. You see, in points I have written the avast for pathogen like this, then the vector, then the symptom. Vomiting is not given in your NCERT, so don't go behind it. Okay, children, are you enjoying this series? Do you think that this series is really helpful for you? Do you think that we are putting our efforts in there? If you think that, definitely tell us in the comment section. We would really love to know that. We would also feel motivated when we see all these things. Okay, so it gives us a good amount of motivation because we are here standing for 2 hours and teaching you continuously. It's not even a live session. So it's a recorded version where we are just continuously recording things for you. So these efforts that we are putting, if you also put that effort for us and tell us in the comment section, or you also study and give the answers as of now, it brings lot of happiness. You can never imagine the amount of happiness that we would get. So definitely follow that. Okay, do let us know. So this is the 20th question. This is a 3-marker question. Now going ahead with the next question. Differentiate between active immunity and passive immunity and define allergy. This is 2017 July question, but this question has been repeated in 2023 or 22 also. Okay. Now, active and passive immunity. I already told you what do you mean by passive and what do you mean by active. Active is when your body is actively producing some antibodies. Your body is actively participating. Okay. Whereas passive is when you are providing antibodies specifically. For example, in the case of vaccine, or probably, for example, I get a disease. When I get a disease, my body will be producing antibodies. Okay, my body is doing that. I am writing my body will be producing antibodies, but this process is going to be very slow. This process is going to be slow. So this is a sad part of this particular thing. Okay, I am talking about active immunity, but there is a good part also. So this is the primary response that it is showing. It will provide antibodies, but this is what the primary response. And in primary response, I told you antibodies are produced. Apart from that, certain memory cells are also produced. Memory cells are also produced. What happens to these memory cells? Basically, they will remember it. And the next time I get this particular disease, this time again antibodies will be produced and it will be a bit more faster. Eventually, the next time I will get this disease, again antibodies will produce. So can I say, the one good thing about this particular thing is it is giving a permanent solution. It is not temporary. It is what? A permanent solution to my problem, because anyways the antibodies will be produced in the first response and also in the second response, eventually in the third response, or whatever time it comes. Okay, initially it was slow, but I made these 2 cells. Next time the secondary response, the antibodies will come in action and will help me out. So this is a permanent solution that I see in the case of active immunity. But when I talk about passive immunity, things are kind of different. Okay, the only good thing is that I am giving ready-made antibodies. Sorry, I am getting what? Ready-made antibodies. And ready-made antibodies, that means it will be really fast. It will help me. But next time my body encounters an antigen naturally. Okay, for example, a snake bit me again. First time a snake bit me, I gave antibodies. Next time a snake bit me, do you think that the body will be producing antibodies? No, it was given. So basically, I will say the action of it, the time of it was temporary. Temporary basis it helped, but then next time it won't be coming and helping. It won't be of any use there. I provided antibodies. When the antibodies did its role, it's done, it's completed. Now the body is not producing more antibodies as of now, because it has not seen the antigen as in general. Okay, so passive immunity is kind of gives me temporary things. Okay, so initially example of passive immunity is one more thing, that is your colostrum to newborn. The mother is providing IgA antibodies directly. Direct antibody is given, but again the body itself, the newborn itself, have to make their own antibodies later on. So this is a very temporary thing. For initial point, when the baby is chutusa, at that point of time the mother is giving, and that will be showing temporary action. Okay, so it's not a permanent solution. And one more thing, as I said, memory cells are involved over here. Memory cells are being produced over here because our body is naturally producing these things, okay, in the primary response, antibodies and memory cells. Over here I would not be having memory cells, which is why again I won't be able to find antibodies, which is why it is temporary. Got it? So let's read it. Oh, before that, what do you mean by allergy? What do you mean by allergy, my dear children? Allergy means there is over exaggerated immune response. Normally it should be reacting, but our body is overreacting to it. So over exaggerated immune response is what you mean by allergy. And in the case of allergy, which is the antibody which comes to play, children? Which is the antibody that comes to play? It is IgE, allergy. You can remember like that, IgE, allergy. So it comes to play. IgE goes and binds on mast cell. What cells, children? Mast cells. When IgE binds on mast cells, and now on this IgE certain antigen comes and binds. For example, dust and things like that. Your mast cell will be releasing histamine. Your mast cell will be releasing histamine, serotonin and things like that. Now this histamine, serotonin, histamine will be leading to swelling and things like that. Okay, swelling, why? Because swelling means vasodilation. Your blood vessels are dilated so that so many cells can come over here. And what say, if other cells are also coming over here, they will try to save us. That is the use of vasodilating. You want more the cells. You are making way for other cells to come and save. So basically over here, histamine and serotonin are doing their job. But actually these antigens should not be causing too much amount of trouble. These are normal antigens. For example, dust is a normal thing. But if our body is allergic, that means it produces more IgE binds on the mast cells, and hence it will trap all the dust particles and everything like that. Basically the antigens which is there in the dust, the pathogens which is there, not the dust, the pathogens which is there in the dust, and then later release histamine and causes swelling and things like that, which is over exaggerated response. So this is what you mean by allergy. Okay, now since I made you understood this thing, I hope you understood, we will see how you can frame the answer. Okay, so active immunity is the immunity developed due to injection of exposure to infection, or basically I'll just write injection of antigens. Okay, injection of antigens as in the dead and the, as I said, the live attenuated form of it. Just a second. Injection of antigens as in what you do in vaccination. Okay, and exposure to infection, that is the cause of active immunity. Now passive immunity, it develops due to ready-made antibodies. Okay, it develops basically with the help of what? Develops with the help of ready-made antibodies. Now it is slow and takes time give its full effective response. Whereas it shows immediate response. This is fast, whereas this will be what? Slow. It provides lifelong immunity. Yes, because you are making your body remind that memory cells have to be made, and next time it comes, your memory cells will be making more antibodies and it will help out. But it provides temporary immunity. Okay, memory cells are produced for long-term immunity. Memory cells are not produced, which is why it gives short-term immunity. So this is the difference between active immunity and passive immunity. Okay, Active immunity, either you can achieve it from a natural infection, or probably you can achieve it if during vaccination. During vaccination, your body is receiving antigens and you are waiting for your body only to produce antibodies in response. So anyways, these both things can happen in the case of active immunity. Now what is allergy? The exaggerated response after immune system of the immune system to certain antigens present in the environment is known as allergy. Okay, you can write about IgE and also mast cells if needed. Cool, this is a 3-marker question, so 2 mark for this, 1 mark for this. Okay, next. 3 marker. Mention the types of carcinogens with the examples for each. Now I told you there will be a question on what type of things can cause cancer. Causative things, causative agents for cancer. So these causative agents are known as what, children? Carcinogens. What do you call them? Carcinogens. So cancer causing things are known as carcinogens. You have different types. Okay. The easiest one is chemical. Okay. The easiest one is chemical carcinogen. So these carcinogens are chemicals which are probably inhaled or intaken in our body, and these chemicals causes cancer. For example, tobacco smoke. Okay, it goes and messes up with our DNA, and this hence, since there is a mess up in our DNA, it can cause cancer. So this is chemical carcinogen. Then comes physical carcinogen. Okay, physical carcinogen as in, for example, you talk about certain radiations. You have 2 types of radiation. One is ionizing and the other one is non-ionizing. Okay, ionizing as in the ones that will be going into the atom, bringing out certain ions. So it is going to cause a huge damage to your DNA. So in this comes your X-ray. Your X-ray and things like that. Okay, in non-ionizing radiation comes your UV radiations. Your UV ray falls under non-ionizing radiation. Anyway, both of them are going to bring out damage in the DNA if given for a longer run. Okay, for a longer time, if it is exposed, then it will definitely cause damage to the DNA. Then after physical, after chemical comes biological carcinogens. For example, in biological carcinogens, again you have 2 types. One is viral oncogenes. What do you call it? Viral oncogenes. Okay, so there will be virus. Inside the virus, there will be certain genes, which can cause cancer. So what do is, this virus, when they affect us, they give this particular gene to us. In our cells, they put it. Hence, we basically form cancerous cells, and these cancerous cells keeps on dividing. So there are viral oncogenes which are being put in our body. Apart from that, in our body we have certain genes which are known as proto-oncogenes. What are they known as, children? Proto-oncogenes. They are there in our body normally, and they do normal function also. The thing is, if certain mutations happen over here, if certain changes happens in this, mutation happens in this proto-oncogenes, it will lead to formation of cancerous cells. So mutations happening in the proto-oncogenes which are normally present in our body, it will lead to cancer cells. So we have chemical carcinogen, we have physical carcinogen, and we have biological carcinogen. In biological, you have viral oncogene, and then you have proto-oncogenes. So these are normally not going to be actively cancerous. Basically, if their mutations have been caused, then they turn to be cancerous. Okay, in ionizing you have X-ray, in non-ionizing you have UV rays. So over here is the answer. Physical carcinogens, ionizing radiation like X-rays and gamma rays, and non-ionizing radiation like UV radiations. So you have to remember X-ray, and this thing comes under ionizing. This thing comes under this thing comes under non-ionizing. This is very much important. Okay. In chemical carcinogen, you have tobacco smoke and things like that. In biological carcinogen, you have viruses causing cancer called oncogenic viruses with oncogenes. So when they put it, we cause trouble. Then several genes called cellular oncogenes or C-onc. It's very important. Cellular oncogenes, or what you call proto-oncogenes, have been identified in normal cells, which when activated or basically when mutated cause cancer. Okay, cool. Can we go to the next question? Let's see. So now comes the 5 marker questions. So 3 marker questions are also done, my dear children. 5 marker question, you won't imagine this easy question in model came in model question paper. Name the causative agents of the following disease. There are 5 diseases that they give, 1, 2, 3, 4, 5, and they are just asking you for the causative agent, and this could give you 5 marks. Can you imagine? That easy question. The thing is, you have to remember the names. And how can you remember? Just like how you have formula sheets in the case of physics and chemistry or any other subjects for mathematics, you have to make a sheet and you have to write the name of the diseases along with the causative agent. You tell it to your friends, you will remember it. How do you think I remember it? I teach you continuously, which is why I remember it, or otherwise I'll also forget. The same thing is applicable for you also. So let's see. Malaria is caused by, Tell me in the comment section, children, if you know other thing, filariasis, ascariasis, amoebiasis, pneumonia. If you know the answer for this, you can tell me already. It is caused by plasmodium, and you have plasmodium vivax, you have falciparum, whatever. Okay, I'm just writing one. But you have to write the whole thing. Okay. Filariasis, I'll be showing you the answer. Filariasis is caused by Vicariaria bancrofti. It is caused by Vicariaria bancrofti, or you can say Vicariaria malai. Okay, Vicariaria bancrofti, or you have Vicariaria malai. So Vicariaria got bankrupt, you can remember like that, Vicariaria bancrofti and Vicariaria malai. Next, ascariasis is caused by Ascaris. It is caused by Ascaris, which is a worm over here. Vicariaria bancrofti is also known as filarial worm. Okay. Amoebiasis is caused by a protozoan which is known as ant amoeba histolytica. Okay, ant amoeba histolytica. Okay, pneumonia is caused by Streptococcus pneumoniae, or it is caused by Haemophilus influenzae. So these are the names that you have to remember. If you can segregate them into bacterial and things like that. For example, malaria, pneumonia, they are all bacterial disease. Pneumonia can cause by virus, but over here in your book it is just given as, what's it? Bacterial. So malaria, pneumonia, bacteria. Okay. Filariasis and ascariasis caused by worms. Okay, nematodes. Your Aschelminthus. Okay, Aschelminthus or the nematodes. Amoebiasis caused by a protozoan. Cool. So over here you have the answer for the same. I have written it properly. You can refer through it. Okay. Actually, the name is Ascaris lumbricoides, but I have not written because it's not given in your NCERT. I don't want to overburden you. Okay. Works? Chalo, next question. What are lymphoid organs? Write the functions of the following in response to immune system in our body. So what do you think are lymphoid organs? I'll say lymphoid organs are those organs which would be making very important cells, that is WBCs and things like that. So I can categorize lymphoid organs into two. One is primary lymphoid organ, and the other ones are known as secondary lymphoid organs. Okay, one is primary and the other one is secondary. Under primary comes two. One is your bone marrow, okay, and the other one is your thymus. Okay, under secondary comes your spleen, you have your lymph nodes, you have your Peyer's patch, you have tonsils. Okay, both of them, they come under MALT, mucosa associated lymphoid tissues. As in, it is there. We have different layers in our body, right? So innermost layer that you see, okay, that is known as mucosa layer. In the mucosa layer, if you have certain lymphoid organs, you call them mucosa associated lymphoid tissues. Okay, basically tissues or organs, whatever you want to say. So Peyer's patch and tonsils is one of them. Tonsils of you have it over here. Peyer's patch you will be having in the intestine. Cool. Anyways, it is associated with the mucosa layer. Now, what is the function of primary lymphoid organs and what is the function of secondary lymphoid organ? Primary lymphoid organ will be the one that will be helping with production of immune cells. So in the bone marrow has certain stem cells which will be giving rise to RBC, that is red blood cells, WBC, and then you have thrombocytes, that is platelets. So all these cells arises in the bone marrow, my dear children. After that, RBC and all gets matured over here only. In WBC also you have different types. In WBC, they're basically leukocytes, you have lymphocytes, you have neutrophils, you have basophils and things like that. In that, okay, in WBC, I'm writing it over here. In WBC, one type of cell is lymphocytes. You have so many others, okay. In lymphocytes, you have B lymphocytes and you have T lymphocytes. B lymphocytes are the one that produces antibodies, children. T lymphocytes basically directly kill the cells. They either directly kill the cells, so in graft rejection you use T lymphocytes, or basically they help the B lymphocyte in finding the pathogen and then producing the antibodies. But anyways, B lymphocytes are the one that produces antibodies. T lymphocytes are the one that basically kills or basically recognizes or basically helps the B lymphocytes. Anyway, you need T lymphocytes. When B lymphocytes do their job, you call this humoral immunity. What do you call it? You call it humoral immunity, because basically this immunity is passed with the help of humor. That is basically antibodies and everything is present in the blood, and blood is a humor. Okay, blood is fluid, which is why you call it humor. So blood and everything will be having antibodies, which is why we say that B lymphocytes provide humoral immunity. Okay, T lymphocytes, we say it provides cell mediated immunity, as in the cells are involved over here in the case of immunity. These cells go and react with other cells, releases something, and so many things happens there. So T lymphocyte with cell mediated, B lymphocyte with humoral immunity. Why am I saying all about this? Because, my dear children, WBCs, I said one type of WBC is lymphocyte, others you have neutrophils, basophils and things like that. Basically, over here, the B lymphocytes mature in the bone marrow itself. But T lymphocytes, it will go to the next area which is known as thymus. So T lymphocytes will be moving to the thymus. And where is thymus? Behind the sternum. Over here behind the sternum you have a small thing that is known as thymus. When we are kids, it is bigger in size. When we become bigger, basically we age, it will become smaller. Okay, so this is where the T lymphocytes, they mature. Rest of the things, they mature here itself. So you got to understand one thing, the primary lymphoid organs are the ones that will help in production as well as what? Maturation. Production and maturation of what? Immune cells. Now what do secondary lymphoid organs do? Secondary lymphoid organs are the one which is acting as a war place. As of now, these guys got produced and got matured, but they have not seen, they have not met any pathogen as of now. They have not started doing their action. So they will be meeting all the pathogens over here, children. They will be actually doing their function over here in the spleen, in the lymph nodes, in the Peyer's patch, in the tonsils. Okay, so you have different organ at different locations. For example, I anyway spoke about bone marrow and thymus. Let me talk about spleen and lymph nodes. So spleen is basically near your stomach itself, you will be finding the spleen. Okay, and the spleen, basically it is known as the graveyard of RBC. It is known as the graveyard of RBC. Over here, the RBCs which are after 120 days, their life cycle gets completed. At this point of time, they will come die over here. Okay, there will be certain cells which will be taking away everything and then killing it in general. Okay, this spleen is also richly supplied with WBC, such that in the blood, if any pathogen comes in, basically these WBCs will capture them and kill them. Okay, lymph node also ones, basically lymph nodes, if you have seen lymphatic vessels, from here the lymph passes. Okay, lymph will be having WBC again, it will be having certain pathogens, it will be having plasma, and then you will be having lymph nodes which are like swellings over here in between the lymphatic vessels, and these are supplied with again too much amount of WBCs. So if pathogen is coming through it, it will catch hold of the pathogen and kill it. Okay, so this is where the actual encounter comes. The spleen, the lymph nodes, the Peyer's patch, the tonsils, it is where the actual encounter of the pathogen with the WBCs happen. They actually do their work. So which is why the doctors, when you go there, doctors, they check over here. If your lymph node has getting swollen, that means infection has happened. Your fight is happening over here. Got it? So I have basically told everything, but let's see the written format also. So the primary lymphoid organs are bone marrow and thymus, where immature lymphocytes differentiate into antigen sensitive lymphocytes. Okay, where the immature ones are going to differentiate into antigen sensitive lymphocytes, as in if anything foreign comes, they will be sensitive to it. That's what they mean by it. Okay. After maturation, the lymphocytes migrate to what? Secondary lymphoid organs. After once it is matured over here, it will move to secondary lymphoid organs like spleen, lymph nodes, tonsils, pairs, patches, or small intestine and appendix. The secondary lymphoid organs provide the sites for interaction of lymphocytes with what? Antigen, or basically the pathogen. Anything foreign comes to be antigen. Okay, which then proliferate to become effector cells. So now the particular cells will be effector cells. There will be lot of things happening over there. So this is what you mean by lymphoid organs. This is the first question. What are lymphoid organs? So you have to talk about primary lymphoid organs and secondary lymphoid organs. This is the most concise way you can say. So primary lymphoid organs are what? Bone marrow and thymus, and this is where the lymphocytes produce and mature. Okay. Secondary lymphoid organs where they actually get exposed to antigens. This is where they start showing their action. Examples are this. Khatam. Done. Cool? Now, bone marrow, the primary site to produce hematopoietic stem cells and then later give rise to RBC, WBC and platelets. Thymus, an organ where T lymphocytes mature. Thymus is active in childhood and early adolescence but shrinks after puberty. Where is it located, children? Behind the sternum. Behind the sternum or the chest bone. Okay, spleen, an organ that filters blood, removing old or damaged red blood cell. It also helps in immune response by storing white blood cells and acting as a site where immune cells can detect and respond to pathogens, just like what I said. Lymph nodes, small bean-shaped structures that filter lymph fluid that circulates through the lymphatic system. They trap foreign particles such as pathogens and are key site for immune activation. Okay, so this is exactly what I said. 2 of them are primary, 2 of them are secondary lymphoid organs. Works? Okay. Chalo. And bone marrow, if you want to understand its structure, in every bone, you will be having a red marrow, my dear children. Red marrow, which will be having these stem cells, which will later form RBC, WBC and things like that. Later the red marrow will be turning into yellow marrow, which will help in making more bone cells, fat and things like that. Okay, works? Okay, next question. Explain different techniques which help in cancer detection and diagnosis. I told you cancer comes in different form, in different question formats, so you have to know all about it. There is no option to leave this. Okay, and we have 3 more questions after which we are going to complete the whole thing. So please don't go run away, listen to me carefully, just be patient with me. Okay, so different techniques which help in cancer detection and diagnosis. Let's see. Okay, I'll teach you from here itself. First technique is biopsy and histopathology. Biopsy, listen to me carefully rather than reading it, which is why I do not show you usually, but okay. Biopsy, for example, I say that there is a cancer tumor over here. I take this tumor and I cut a portion of it. This process of removing a portion of it, it is known as biopsy. Now, after removing, I have to check, right? This process, when I am going to check this particular thing, it is known as histopathological studies. It is known as histopathological studies. So, in biopsy, a piece of the suspected tissue cut into thin section and is stained and examined under microscope. This is known as examination is known as histopathological studies. So, this is one technique to diagnose things. Okay. Second thing, imaging techniques. So, we have computed tomography, which is also known as CT scan. Okay, in CT scan, what do you use? You use X-rays to generate a 3-dimensional image of the internals of an object. So, you get a 3-dimensional with the help of X-rays only. You are going to have a 3-dimensional 3D image of the internals of an object, or basically your body parts. Apart from this, you can use normal X-rays also, which again gives you which helps you detect things. But the problem with this and this is that it is using ionizing radiations, and it's not really good for our body. It's not really good for our body. So, you say in imaging techniques, first thing is you do what? X-rays. Second thing, with the help of X-rays, you do CT scan, that is computer tomography, where you are giving a three-dimensional image of the internals. Third thing, which is the most good option, that is MRI, which is why it is costly as well. Here you are not using ionizing radiation, instead you are using strong magnetic field and non-ionizing radiation, okay, to accurately detect pathological and physiological changes in the living tissue. So this is the best out of these three. Okay, they all come under imaging techniques. Fine. So one is biopsy and histopathology, then comes imaging techniques. For that you have X-ray, then you have computer tomography, then you have MRI. Now, antibodies against cancer specific antigens, that is also there. So basically what you can do is you can collect a sample. For example X person. Okay, X person, I am supposing that this person has cancer, but I am not sure. What I can do is take their blood sample and then put it in a well. Okay, now what I am doing is I have already put some antibodies over here, which is very specific to antigens, which basically cancerous antigens, okay, which are very specifically produced in bodies of organism or basically animals or human beings who have cancer. Suppose a person had cancer, I took certain things from them, the antibodies which are released in them, okay, when they had cancer, and I have kept it. Now I am using these antibodies to detect. When I am putting the blood sample, if the antigen is going to bind on this antibodies, that means this person is also having what? Cancer, which is why it is getting bound to these antibodies, which are very cancer specific. So this is also a way. So antibodies against cancer specific antigens are also used for detection of certain cancers. So you're having the antibody which is being produced in response to cancer specific antigens, and now when you're testing the blood, you see that the organism, a human being that you are taking, it is also going on attaching. The antigens of that particular being is also going on attaching to the antibody. That means, yes, it is what? Cancerous antigen. So that person has cancer. Okay. Now apart from all this thing, techniques are there of molecular biology that can also be applied to detect genes in an individual with inherited susceptibility to certain cancers. So there are, for example, breast cancer. You can see female to female, it goes on continuing from grandmother to mother to daughter, it continues. So if we have already certain techniques, certain molecular biology techniques with the help of which we can identify those genes. Okay, and if it is there in other organism, that means yes, that organism is also susceptible to cancer, and then we can probably do something about it. Yeah, that is also possible with the modern age molecular biology. So these are the ways where you can diagnose cancer, my dear children. Works? Chalo, next question. Easy question. This is an easy question. Some drug bottles had their name label missing in a drugstore of a hospital. The staff needs to identify the drugs with their actions still written on them. So the drug list is given, the effect is given, but the name is not there. You have to label it. That's the task. Let's get started. So it is used by doctors as sedative and painkiller. You tell me, we already studied this. What is that drug which is already being studied? Over here, it is used by doctors as painkiller and sedative. It will be morphine, and morphine comes under opioids. Okay. Next thing, helps patient to cope with insomnia and depression, as in people who are not able to sleep or they have depression. This particular drug is used for that, and I don't think you might know it now, or if you have already learned the NCERT, you might know it. Do you know benzodiazepines, amphetamines, things like that? I will show you. I am not writing it over here. I will be showing you there. It will confuse you or else. Okay. So let's keep it as of now like this. We will discuss it. Increases blood pressure and heart rate of consumer. Nicotine, which is there in the tobacco, it basically increases the blood pressure and heart rate of the consumer who is consuming this. So this is one other drug. Okay, drug means what? All the chemicals basically that is being used. Now acts as depressant and slows down the body function. This is again depressant. We learned heroin. Heroin is going to act like a depressant. Affects cardiovascular system of the body. I told you there is only one thing that affects the cardiovascular system. It has to be cannabinoids. cannabinoids. Example, ganja, marijuana and things like that. Rest, opioids as well as coca alkaloids, cocaine and stuff like that, they are going to affect your CNS. But over here they are affecting the cardiovascular system. Now let's check the answer. So over here we have morphine, and as I told you, the examples, amphetamines, benzodiazepines and barbiturates are examples of examples of drugs that can be given to people who have insomnia and depression under the doctor's surveillance. Okay, not that you are going to give them. Okay, it should be done under doctor's surveillance. Barbiturates, amphetamines and benzodiazepines. What do you call it? Benzodiazepines. Amphetamines, benzodiazepines and barbiturates. Okay, nicotine, blood pressure, depressant, heroin, cannabinoids, cardiovascular system. Okay, this is the only one that affects on the cardiovascular system according to your NCERT. Cool? Done? Okay, got the 5 marks? This came in the model question paper, so you have to better learn it. Okay, next. Okay, what are the measures taken to control alcohol and drug abuse in adolescence? So what are the steps that are taken to control this thing? Can you tell? Easy things, can you tell? This is something that is theory based. This is not something that you can remember. You have to remember. It's very easy. What things can be done? Don't give undue peer pressure. Okay, talk to your friends. These are the things that you can do. Seek professional help. Okay, so if you want to know the answer, it goes like this. This is directly taken from your NCERT. So prevention and control measures for drug and alcohol abuse, avoiding undue peer pressure, because when you give peer pressure, it will obviously create a lot of trouble. They might go take drugs and things like that. We want to prevent it. Education and counseling. The child should be educated and counseled to face problems and stresses and to accept disappointments and failures. It should be encouraged to take up healthy hobbies like music, reading and sports. And I hope my children, all the Chutku people, they are very well aware of this. That basically problems have to happen. This is life. This is the only life you get. So without problems, what's the zeal about? Okay, so you will have problems. Face the problems. That's the challenge that you get every day. So there is someone who is challenging you every day. Remember like that. And you have to win in this challenge every single time. Okay, and sometimes you might lose, sometimes you might lose, you might feel sad. It's totally okay to feel sad, to feel gloomy. It's totally fine. Okay, it's an emotion and we are the human beings who have all the emotions. So it's totally justified to be sad. But please remember, the day is not over, the world is not over. It still has something to offer you. Just wait for that. Be curious to find what is there still left for you to find out. Okay, so education and counseling has to be given, and then seeking help from parents and peers. These are your best friends, my dear children. Your parents and your peer would be best friends, so talk to them. They'd help you a lot. Okay, looking for danger signs. Parents, teachers and friends should be alert to identify danger signs associated with drug, alcohol abuse, so that appropriate remedial measures of treatment can be initiated. So now if you think, you know what, I had my friends. Okay, when I was in school, I never tried a lot of things. Okay, I never tried, because I didn't want to be that cool like others. So the problem between children is that it has happened every time. They think that some people who do not try are not cool, and the people who try, they are very cool. It does not go like that. Please understand the fact that that is nothing to be cool. And what are you achieving after getting that coolness award? Is there something that you're achieving among your friends? Nothing, my dear children. So please understand, if you're getting hang of it, you will keep on doing it, and we do not want that. Okay, so basically in a world of people who are fighting for coolness, be that person who want to be a change. Okay, that's what I would definitely say. That's more challenging rather than to be cool. To be like everyone else, it's very easy. Just be yourself. Do not try all these stupid things. It will be really, really bad for your health. Okay, at one point of time you would realize it after growing up, and that's kind of common. You and me do not have very much age gap. It's not like me being a very big oldie is explaining you. No, I'm telling you with my experience, and please count on me for that. Okay, so that is one thing. Now, seeking professional and medical help. Affected individuals should seek the help of qualified psychologists and psychiatrists, and also de-addiction and rehabilitation programs to come out of the problem. Yes, make it really, really common thing to go to a psychologist to talk to them. I had children coming to me and talking about a lot of things. When I was not able to handle things, I used to send them to other people who were actually trained for this. Obviously, I'm just a human being who have no knowledge about things, okay, much things. Emotionally, I can support to one level, but obviously after a point of time, there are things that other human beings cannot do. people who have good learning about this criteria and things like that, they could help you better. So you should be seeking professional and medical help if you are in this particular trap. Works? So it's all cool to do all these things. Please understand this very important fact, and this is the measures that can be taken to control alcohol and drug abuse in adolescents. Cool? Works? Now the last question we have for the day is this. Give the schematic representation of the replication of retroviruses. So how does retroviruses do their action? I just want to tell you once and then I'll go ahead. So let's take a retrovirus, that is HIV virus. Okay, suppose this is a cell. Okay, just give me a second. This is a cell, this is the nucleus, and this is our DNA. Okay, there comes the virus which has its capsid layer, okay, and things like that, proteins. It will be having its own genetic material. This virus will come and bind over here. Okay, suppose it came and bind over here. It sends its DNA inside. Sorry, not DNA, genetic material inside. It sends its what? It sends its genetic material inside, whatever it can be. So basically I'll take HIV only. HIV has 2 RNA like this. Okay, HIV has 2 RNA like this. So here also I'll make that. It has 2 single-stranded RNA. They are not coiled to each other, they are single-stranded RNA. And apart from this, they have 3 very important enzymes. They have 3 very important. So when the cell comes, basically it gives these enzymes as well and the RNA. Now, the first enzyme out of this is known as reverse transcriptase. Okay, and if you remember what is the role of reverse transcriptase, it was making DNA from the RNA. So here also it will make DNA out of the RNA. So let's make a DNA out of it. Suppose it made the DNA. Okay, suppose it made the DNA. Okay, the next thing it want to do is basically integrate this DNA into the DNA of ours. So the next enzyme that comes to play is known as integrase. And what does it do? It basically lets this particular strand which is being made integrate into the human genome. Now over here we have the viral genome and this is the human genome. The human genome does not know that there is an extended form over here. It will keep on replicating, it will do transcription, it will make mRNA out of it, and this mRNA will make protein out of it. Lots of things happens. So over here what happens is what? This guy will be making viral mRNA. There will be viral mRNA being formed. Okay, and this viral mRNA will be forming peptides or basically proteins. And to make them active, you need another enzyme which is given by the virus only, and this is known as, where is it? And this will be known as protease. So protease will be basically breaking down the proteins into smaller fragments such that you can again make, so these proteins that you see or these enzymes that you see is nothing but the proteins itself. Enzymes are just nothing but the proteins. So basically with the help of viral mRNA, I made the proteins that I needed. Okay, and I also have my RNA strands. I also made my RNA strands and everything. Okay, so basically can't I just make a new virus out of it? I'll make a new virus out of it and this will go out. This will go out, children, and it will infect other cells just like this. So basically it is using our machinery to affect things. So in the case of HIV virus, the cells that is initially affected is macrophage. You know the function of macrophage? Macrophage takes in other organism and kills it inside. So macrophage basically takes in this particular thing, but what happens to macrophage is that it's not that it takes in things. Basically there are receptors on it, okay, in our cell, and these receptors bind with the proteins over here, and then it takes it in, and then it uses our machinery itself and makes its own copies. And then after leaving the macrophage, it will be going and affecting T lymphocytes. And you know T lymphocytes is the one that saves us from everything. And this time if you're going to affect T lymphocytes, what will happen, children? Your whole immune system gets damaged, which is why you will be having so many problems. So T lymphocytes, in T lymphocytes, what are the ones that initially gets affected? T helper cells. So I told you, no, there are certain lymphocytes in T lymphocyte which will help the B cells or B lymphocytes to produce antibodies. It is the T helper cells. So T helper cells are the one that comes in an intermediate thing. Just like a friend that you have. For example, in 3-friend group, there will be always one friend which will be solving all the problems. So similarly over this TH cell is the one that helps always in all the immune response. But if the virus is going and acting on this TH cell, don't you think that will be a problem? Yes, it will be a problem. So please remember, this is how things work. This is how the schematic representation of the replication of retroviruses takes place, but I will show you a different diagram also. This was just for your understanding. Okay, so it is human immunodeficiency virus, that is HIV, causing acquired immunodeficiency syndrome, which is known as AIDS. And this is the enzyme RIP, reverse transcriptase, integrase and protease. It is causing RIP for our cells. And this is how it actually looks. So this is a retrovirus. This is a viral RNA core. Okay, there is RNA over here. Wait, I'll just zoom in and show. Okay, viral RNA core is there. The virus infects the normal cell over here, you see, and then RNA is introduced over here. Viral DNA is produced by reverse transcriptase. You got the viral DNA. This viral DNA incorporates into the human genome. New viral RNA is produced by the infected cell. It will only make it. New viruses are produced. Apart from new proteins are also being produced, and then later it moves out. So this is how you can explain. Okay, you will get this PDF if you join the, if you fill in the form. And basically if you fill in the form, we will be giving you through your WhatsApp only. You don't have to join anywhere. Okay, so fill in the form which is given in your description box. This is a very important thing. Okay, then you will get all the notes. You can copy it from the notes, or probably you can just remember it with the help of this also. Works. So with this we have completed all the 28 questions, and as you can see, the most important topics from this chapter will be the diseases, the causative agents, everything, the symptoms, everything that you can totally find. Apart from that, active and passive immunity is important, allergy is important, lymphoid organ is important. Okay, active and passive immunity, lymphoid organs, and what? Lymphoid organs, and then diseases, drugs, cancer, very much important. Okay, we just left for treatment, so just go through the treatment also, the chemotherapy and all that once. Okay, and apart from it, what I think that's all. That's the most important thing. Just go through it. And HIV is also important. Okay, so that's all. I hope you just have a quick read of NCERT as well once. Okay, it will just cement things in your brain. This video is totally enough for your preparation. I have put all the model question papers, or basically model questions, which have come from this particular chapter over here in this particular video. So it's all completed within almost 1 hour 32 minutes, and I think it's a good deal. Right, you can put it into X speed and listen to it whenever you want, but definitely refer to this whenever you need. Okay, so that's all, my champions. Now I am also kind of a bit tired, so we will stop it over here. And if you have any doubts, okay, I would love to help you in there in the comment section. So put down your doubts in the comment section. Tell me if it was the video that you think over here was useful or for you or not. As I said, it always boosts us, so do not forget to do that. And I wish that you practice lots and lots of questions, okay, apart from this also, for every chapter, for every single subject as well. Later on, as of now, you can go through this. If you have time later on for revision, then definitely go through your NCERT or solve more questions whenever possible. So that's all. If you think that this particular video was helpful for you, do not forget to subscribe to our channel, and then take care. Bye-bye.